technique known as centrifugation. Now, before we proceed any further, let us introduce ourselves. Hi again, my name is Jinan. In this video, we'll break down how a centrifuge works, the key parts of the machine, the basic steps to run one, and how to interpret the results. Let's go! Centrifugation is a technique used in laboratories to separate components of a mixture based on their densities by spinning a sample in a centrifuge machine at high speeds. The centrifugal force, which is caused by the spinning motion at high speed, will cause heavier particles to sediment at the bottom of the tube, forming what is known as a pellet, while the lighter substances will remain in the liquid above, forming what is known as a supernatant. This technique is especially important in biochemistry because it allows scientists to isolate cells, organelles, proteins, and nucleic acid. By separating these components, researchers can study their structure, function, and chemical properties much more accurately and easily.
Continuing on, a centrifuge is a lab instrument used to separate components of a mixture based on differences in density by applying centrifugal force. When the rotor spins, particles in the sample experience an outward force away from the axis of the rotation. This force depends on particle mass, rotational speed, and distance from the rotor center. It is commonly expressed as relative centrifugal force, RCF, which measures the effective force on the sample relative to gravity and rotor radius. Denser particles experience a higher RCF, move outward faster, and form a pellet at the bottom of the tube. Lighter particles remain suspended in the liquid phase as the supernatant. In short, centrifugation accelerates sedimentation, allowing rapid and controlled separation. By adjusting RCF, centrifugation time, and rotor type, specific components can be selectively isolated. Basically, a centrifuge separates particles by exploiting their density difference under controlled centrifugal forces.
The lid ensures safety by locking the samples inside and preventing the centrifuge from operating when it is open. The control panel allows the user to set the speed and time required for proper sample separation. These components work together to ensure efficient and safe centrifugation.
How do we operate a centrifuge? The method is simple. We just need two things, which are a centrifuge and a beam scale. The centrifuge is the workhorse for our job, while the beam scale ensures equal mass between two tubes containing sample to prevent damage to the centrifuge. Before using a centrifuge, the tubes must first be balanced until they are of equal mass. This is the purpose of a beam scale. Simply place two tubes on each pan and observe if they are of equal height with each other. If one is lower than the other, use a droplet to take some of it and transfer it into the other one until they share the same height. Finally, to use a centrifuge, place the two tubes inside the centrifuge tubes oppositely to balance the rotor. It is important that one tube must have its counterpart and both are placed oppositely with each other to avoid damaging the centrifuge. To adjust odd number of tubes, simply fill another tube with distilled water, balance it, then place it into the chamber.
When the centrifuge comes to a stop, we can observe the result. There will be two different layers. The top one is called as supernatant, meanwhile the bottom one is called as pellet. Usually, the heavy molecules will stay on the bottom layer, meanwhile the lighter molecule will stay on the top layer. Now, by changing the RCF value, we can target different components. Higher RCF values means that the pellet form will be much lighter. Meanwhile, a lower RCF value means that the pellet form will be much heavier. That's all from me. Thank you.
In conclusion, centrifugation is a vital and widely used technique in biochemistry that allows scientists to separate biological components efficiently using centrifugal force. By spinning samples at higher speed, heavier particles will move to the bottom of the tube to form a pellet, while lighter substances remain at the top of the tube as the supernatant. Throughout this video, we have explored the basic principle behind centrifugation, identified the main component of a centrifuge and their functions, and reviewed the correct operating procedure to ensure accurate and safe results. Centrifugation plays an important role in many biochemical properties, such as isolating cells, separating organelles, and purifying proteins. Without this technique, many laboratory experiments and analyses will be much more difficult to be and less precise. Therefore, understanding how centrifugation works is essential for anyone studying or working in the field of biochemistry.